Institute is the smallest BBSRC-funded institute, but on the contrary to being the smallest, I think it's one of the most diverse institutes.
People do really broad work at the Earlham Institute.
So we work on polecats, plants, down to tiny microorganisms across all different parts of the tree of life.
My name is Maddy Allison. My PhD is all about identifying plant diseases in the air by capturing their DNA and sequencing it. I knew that I wanted to be in the field of food security or DNA sequencing and bioinformatics, so that was my main focus. And then I just came across the AirSeq project, and it just seemed really exciting because it's something I'd never heard of, DNA in the air. So it's sort of like a really big vacuum cleaner, so it's sucking in all that air which becomes trapped on the filter in the middle of the sampler. And then we'll take it to the lab to do the sequencing part.
Hi, my name is Melissa Salmon. I'm a postdoc researcher working in the Earlham Biofoundry. So I'm working on a project that's part of a large consortium of researchers. What we're aiming to do is to build a synthetic plant artificial chromosome. And the aim of this is to be able to introduce beneficial traits into potatoes, so things that can improve nutrition of the potato or disease resistance.
My name is Edita Vujtovic. The area of research that my group is interested in is stem cells, in particular blood stem cells, to refine existing therapies and improve current diagnostic methods to improve human health. So here we see the cells that are producing platelets. Now, if you think about analyzing specimens from patients, the first line of screening is visual inspection and counting cells. Now, time is working against us, so the sooner we know the answer, which combination of treatment we should administer, the higher the chance of the recovery. Very recently, we have obtained a sequencing data set where we have molecular information about these cells combined with their images of cells. Now we can start linking the image of the cell with the molecular properties. It may speed up the process of diagnosis.
Technologies have massively advanced what we can do. So we can do things faster, everything is a little bit easier, and that really accelerates the speed that we can get results.
The institute is small, which means we know each other.
It's kind of a fascinating place to work. We work across a whole wide range of different areas of science, different disciplines.
Having an interesting conversation with one of your colleagues, it ends up as a start for collaboration. We can combine different types of expertise in the same project to have more holistic understanding of specific processes.
Being in a research environment and learning all these skills makes me feel like myself and other young people can really make a change and a beneficial impact on the world.
So the ultimate aim would be to be able to introduce synthetic DNA that would be able to provide traits that are going to help food security to be not such a big issue in the future.
In like a dream scenario, it would be amazing if we had different monitors all across the UK or the world to constantly identify what's in the air and build that into models, informing growers of what diseases are present and helping them protect their crops whilst minimizing the amount of chemicals that are needed.
From one hand, it's very challenging because we stumble upon hurdles very often, but at the same time, whenever we achieve a milestone, it's very motivating and it's a positive reinforcement. After a lot of troubleshooting, these things can work.